We can produce RNA-based vaccine, we can produce vector-based vaccine, we can produce immunogen or protein-based vaccine on the basis of the scientific knowledge or the fundamental infrastructure we have. When we talk about vaccine development, we can simply, in simple language, can divide into three phases. The first is fundamental science about the development of vaccine, and then when you develop a molecule or maybe truncated virus or any type of immunogen which is capable of induce immune response as a vaccine, then the second phase is its characterization in animal models. So this is a critical phase. So when all these things are characterized and a molecule is passed, then it goes to human trial. So there are generally three types of vaccine. The one type is called vector-based, then another is called RNA-based vaccine, then the third is truncated virus or heat inactivated virus-based vaccine, where the whole virus is basically get inactivated by the process of heating or chemical modification. But the fourth and the modern type of vaccine is a protein-based, immunogen-based vaccine. This is really the most effective approach. When we talk about our capacity, so we have almost everything of the first step that we can produce those molecules. A protein or immunogen is found as non-toxic. It is injected into animals, and the serum of that injected animal is taken, and then that serum is tested whether it is inhibiting virus or not. So that thing we have already very well established, those neutralization assay. We have made pseudo-viruses of coronavirus. Pseudo-viruses mean that the virus is just like original virus. It has its genome truncated. It means that it will infect cell only once and it will not produce more, it will not replicate because its genome is half truncated. So we have produced pseudovirus of COVID-19, and if a vaccine, a potential vaccine is given to animals and animals create antibodies, we can test those sera whether how effective that vaccine was. So we have those type of capacity here.
Now at LUMS we have this capability. We are designing and in some cases have designed constructs which will produce this spike protein as a proof of concept in our case. Here we will also use our indigenous strains which are targeted for our local population.
I think this is a wonderful initiative, and it's really great opportunity for all of us. I think we are capable and we are interested in contributing in this project in several ways, by characterization of vaccines at different level, by evaluating its effect, by evaluating it preclinical phase, and testing in the lab whether that serum is inhibiting or neutralizing the virus. So we can provide that kind of contribution to evaluate a vaccine when it is produced.